Hello, everyone. Okay. Hi, Mike. Hi, James. Who else?
Hello.
Hey.
Hi.
I guess I could mute myself. I'm sorry, guys.
You're fine. Okay, so what's the time? Oh, we have 2 more minutes. Okay, so your test, second exam, I think it will open on Thursday, right?
I believe so.
Yeah, I think so. It will open on Thursday morning, Thursday, Friday, Saturday, and it will close. Will close on, I think, Thursday, Friday, Saturday night. So, yeah. Okay, so what I'm going to do is I will try to complete this chapter today. If I cannot, then rest of the chapter and chapter 9 I will record maybe tomorrow, and then I will post positively by tomorrow night. Positively by tomorrow evening before my class, I will post rest of the material. So you will have everything for. Okay, so let's start. This chapter is really huge chapter. I think I need to start from here. You can see my PowerPoint, right?
Yes, ma'am.
What happened? Why can't I? Oh, I think I talked up to this, right? I talked about autotroph,
You did.
heterotroph. Yep. So I
Yes,
think
ma'am.
from oxidation, yeah. So from redox reaction, yeah, I need to start. Okay. So let's start. Let me grab. Okay. So I'm going to open whiteboard, okay? And then if you, when I will come back to PowerPoint, and if you do not see my PowerPoint, let me know, okay?
Yes, ma'am.
Yeah. Okay. So oxidation and reduction, okay? Basically, so let me write over here. So oxidation and reduction reaction. So together we actually called it redox reaction, okay? We have basically two type of chemical reaction that occurs in this type of that inside this oxidation reduction reaction, okay? One type of chemical reaction, it's called oxidation, okay? Another type of chemical reaction that is called reduction. Together, this oxidation and reduction together is called redox reaction. Somebody is joining. Okay. So what happens during this oxidation and reduction reaction? Basically, electron actually moves. electron moves within the elements. Okay, so let's say two atoms here, A and B. Okay, A one atom, B another one. So let's say A donate one electron to B, and B is actually going to accept that electron. So in this case, what happened? Electron is moving from atom A to atom B. So when electron is moving within the atoms or between the atoms, that is a redox reaction. Okay, or you can say oxidation reduction reaction. So two things are happening over here. Atom A is donating, so it is donating and donating the electron, okay? And atom B, it is actually receiving the electron. So two things are happening. So electron is moving in this system, so it is a redox reaction. Now, what happened? If we go by the definition, oxidation means losing the reaction, losing the electron. So when one atom lose electron, that process is called oxidation. Reduction, when one atom actually gain electron, then that process is called reduction. Okay, it has one mnemonic, like oil rig. So oxidation is losing, reduction is gaining. So that's the definition of oxidation and reduction reaction. So I hope you understand that. Okay, so one atom is donating the electron. So there must be someone who is going to accept that electron, right? So that means over here, one donor and one receiver must be present, and that's why every time this oxidation and reduction reaction, they actually go together, and that's why instead of oxidation reduction reaction, what we call this reaction as redox reaction. Okay, so these two reaction, oxidation and reduction, they simultaneously goes together. And the definition of oxidation means the reaction when one atom lose one electron, that's the oxidation. Reduction reaction, when one atom gain or accept one electron, that's the reduction reaction. Okay, so that's the definition of oxidation and reduction reaction. Now few things we have to consider. So over here, just this reaction, A is donating electron to B. Okay, so over here, atom A is donating. Okay, so then based on the definition, oxidation is losing. So atom A is losing one electron. So that means this reaction is called oxidation reaction. Okay. Now reduction is gaining. So over here, when atom B is accepting or gaining this electron, that means over here, this reaction should be reduction reaction. Okay. Now, what happened when atom A donate electron means by the process of oxidation, when atom actually lose the electron, then this atom actually is going to be oxidized. Okay, because it is going under the process of oxidation. Now atom B, when it receives, means reduction reaction occurs. So we can say that atom B is actually going to reduced. So now we can say that atom A is an oxidized agent, okay, and atom B is an reduced agent. Okay, so after the oxidation, that atom or that element is oxidized. After the reduction, that element or atom becomes reduced. Now, remember at the beginning, I told that these two reaction. So one person is donating, so receiver must be there. So these two goes at the same time. So that means this reaction, this oxidation reaction, is actually going to help that receiving or the reduction reaction to be happen. Okay, so this is actually helping to have that reduction reaction. So atom A is actually helping atom B to be reduced, right? So we can say that atom A is actually a reduction agent, that we can say, because it is A actually helping B to be reduced. So that's why it's reduction agent. Like if you want to buy house, what agent does? Agent help you, right? So that's the job of an agent. So over here, reduction agent means the agent who is actually helping some atom to be reduced. So that's why over here, A is reduction agent. On the other hand, B is receiving electron, okay? And it is helping A to be oxidized. So that's why B is oxidized agent. Okay, I hope now you are clear about oxidation and reduction reaction. As long as you can understand that what is oxidation and reduction, and how actually one atom become oxidized and how one atom is is reduced, and why one oxidized agent is also called reduction agent, and why one reduced atom or reduced I don't want to use that agent, I will use atom over here. Reduced atom is actually going to be oxidized agent. I hope now you are clear about that. Another thing I want to tell that, look, when inside a chemical reaction that electron is moving from one atom to another atom, right? We really cannot see that thing is happening, because you can understand that electron, that's a subatomic particle, so we cannot see the movement of electron. But in a chemical reaction, we clearly can see the movement of oxygen and hydrogen. Okay, so oxidation means addition of oxygen. Okay, reduction means oxygen actually basically losing of oxygen, or you can say addition of hydrogen. Okay, or losing oxygen. We can see the movement of oxygen and hydrogen, but we cannot see the movement of electron in a chemical reaction. So by looking at it, or by calculating, when we are actually writing down all the chemical reaction, it becomes very easier for us to just look for the movement of oxygen and the hydrogen. Okay, so that's another definition. But this is actually that oxygen is losing of electron, and reduction is gaining of electron. That's the actual definition. But for do the math in a chemistry lab, or in chemistry, to do the math, we actually look after the oxygen and hydrogen. Addition of oxygen is oxidation. Losing of oxygen is actually reduction. But in chemical reaction, every time what happens, if you lose one oxygen, you are going to add hydrogen. Okay, so that's why addition of hydrogen or losing of oxygen is called the reduction. So both definitions are correct, but this is actually for biology purpose, this is original definition. Can you see my PowerPoint now?
We currently see you on camera, Ms. Lundy.
Huh?
We're seeing you on the camera. We don't see the PowerPoint right now.
Oh, you don't? I don't know why. What? Now?
No, ma'am.
No.
Okay, I'm not going to. I think but I'm sharing the PowerPoint.
We can see your screen now.
You can see it now,
Yeah.
but you cannot see my PowerPoint.
No.
No, currently the background is like the Zoom website. There it is.
Yep.
Now you can see?
Yeah.
Okay. I don't know, but what happened? Now I cannot share it. Okay, let me see if I. You can see it now?
Yes.
Okay. Okay, so okay. Okay, so I hope you got that oxidation reduction reaction. Over here, that's one example. I just went through that, I gave you example like atom A and atom B. Over here, it's talk about sodium and chlorine. So I hope you remember that how we actually draw the electronic configuration. So, and last chapter, I think we worked on the covalent bond. So what happened when sodium, okay, so when sodium and chlorine together makes sodium chloride. So what happened? Sodium has 11 electron, okay? Chlorine, it has 17, okay? So, and this is the electronic configuration for sodium, and this is for chlorine. So sodium has 2, 8, and outermost layer, it has 1 electron. Chlorine, outermost layer, it has 7 electron. Based on the octet rule, I hope you remember that atom has a tendency to get 8 electron on its outermost layer. Okay, so the sodium, what it will do, it will actually get rid of this extra electron. So if it donates this 1 electron, then what will happen? Then the next orbit, that is the second orbit, will have 8 electron, and it will be happy. Okay, it will be fulfilled. Now chlorine, it has outermost orbit has 7 electron. So if chlorine accept this electron, then outermost orbit of chlorine will be 8. Now if they do that, then both the atom will be very happy, fulfilled, and they will be like, you know, chemically inactive, and that's the target of the atom always. So in this case, sodium is going to donate 1 electron to chlorine, and chlorine is actually going to accept that electron, and our definition is oil rig. Oxidation is losing, so oxidation means losing. So over here, sodium is losing electron, means this is the process. So when sodium is donating electron, and it is going to be in A plus, so electron has charge negative, one negative charge. So if it lose 1 electron, then it converts into sodium ion. It's not an atom anymore. Okay, it's an ion. So sodium is donating 1 electron and converts into sodium ion. That process is called oxidation. Okay. Now over here, chlorine is gaining 1 electron, means chlorine is gaining 1 extra negative charge. So chlorine is actually going to convert into Cl minus ion. Okay. This process, when chlorine gain 1 electron, okay, and it converts into negative ion, so that process is called reduction. And these two need to go, this reduction, this process, and oxidation always goes together. Now after oxidation, what will happen? When Na converts into Na plus, after oxidation, that means sodium is going to oxidized. Okay, and over here, chlorine is going to be reduced. Okay, I hope this up to this is clear. Now sodium is helping chlorine to be reduced, so sodium is called reducing agent. Now chlorine is helping sodium to be oxidized, so chlorine is called oxidized agent. Okay, it's pretty simple. Okay, electron carriers. So electron carriers main, actually basically this chapter, our main focus is on the metabolism, means how we are going to break down glucose and produce oxygen inside our body, sorry, carbon dioxide and water. Okay, and produce ATP. That's our main focus in this chapter. So when actually glucose breaks down into carbon dioxide and water, what is happening over here? You can see that when it was glucose, so it had 6 carbon, 12 hydrogen and 6 oxygen. So it is a 6 carbon compound, okay? So from there, it breaks down into just 1 carbon compound, okay? It becomes small inside. So this process occurs in the presence of oxygen, and basically what happens, carbon over here directly is going to add up with the oxygen. You can see over here. So in this case, if you break down the C6H12O6, what happens, that hydrogen is out, okay, and oxygen is in. So basically, we can say that this is the process of oxidation. When glucose is breaking down into carbon dioxide, so that's the process of oxidation, because glucose is going to lose the hydrogen. Okay, so remember that addition of oxygen is oxidation. Means addition of oxygen, if addition of oxygen is oxidation, then losing of hydrogen is also oxidation. Okay, so over here, this is the process of oxidation. Now here, that ATP, glucose, this oxygen, they are the you can tell that they are the heroines of the main scene. Okay, now these electron carriers, they are actually behind the scene, actors and actresses. Okay, so this is the main thing, but main event that glucose is going to break down into carbon dioxide and water. Okay, so glucose is taking part in it, producing ATP. So all those are very, very important. Okay, but how it is going to happen, like breaking down or oxidation of glucose into carbon dioxide to happen, there are lots of other elements or compounds or molecules, you can say, are there who actually help glucose to transform into carbon dioxide, and they are the electron carriers. Okay, so what happens, so when actually basically this thing happens, like C6H12O6 breaks down into carbon dioxide, or you can say that oxidation happens. Okay, what happened, this is not just the one step. Okay, it happens in so many different steps. Okay, so basically it is, you can say, like it's like a staircase. Okay, there are multiple stairs there, through which that glucose starting from here ultimately produce carbon dioxide here. Okay, and so many multiple chemical reactions are there. So this is one reaction. So over here we have one end product. So over here we have another end products, and every time what happens, when that glucose breaks down into something, like let's say over here the production of atom A, over here production of atom B. When this is happening, so from glucose to A to B, B to C, over here every step they lose electron. Okay, now this electron, what happens, this electron, there are few you can say that that the shuttle bus are there, who actually carry these electron. Okay, so where do they carry? They carry electron to the electron transport chain. Okay, um, where by utilizing these electron, cell ultimately produce that 34 to 36 ATP. Okay, so all these electrons, so from here to go over here inside the electron transport chain, that's very important step. You know, if electron does not reach over there, then glucose will not be carbon dioxide. So to do that, there are few shuttle electron carriers or electron shuttle are there, like NAD, FAD. They actually carry electron from here to basically they carry electron into the electron transport chain during the metabolic process of breaking down glucose into carbon dioxide. Okay, so these electron carriers, they are very important for our cellular respiration. NAD or NADH, NAD, the nicotinamide adenine dinucleotide, very important for photosynthesis for plant. There is FAD, flavin adenine dinucleotide. So FAD converts into FADH and NAD converts into NADH. So these two are very common electron carrier in for our metabolic pathway. So over here, NAD converts into during cellular respiration. This is very important. You have to know that. Like during cellular respiration means when actually glucose breaks down into carbon dioxide in the presence of oxygen and produce ATP. That's actually called cellular respiration. Okay, so what happened during that time? This NAD, FAD, they actually carry the electron, right? They are the electron carrier. So NAD converts that accept electron and they converts into NADH. Okay, so when NAD donate one electron, it converts into NAD plus, right? And it ultimately converts into NADH. So that NAD and NADH, when this, so these are the two form. Okay, one is oxidized form, or you can say NAD plus and NADH. These are the two forms. Okay. NAD plus, that's the oxidized form. You can understand that when it's ion, means it's donated, and plus ion means it's actually donated electron. So that's the oxidized form. Over here, addition of hydrogen, that means this is the reduced form. So this is very important that you can identify the oxidized form and the reduced form of any molecule or compound. I will give you one chemical reaction during the test, and you have to identify that which one is oxidized and which one is reduced. So very easy. Make sure that electron is going to donate. Addition of oxygen or reduction of hydrogen. These are the three criteria to be oxidized. Okay, this is reduction. So if you remember these three, then it's very easy to identify. Opposite, addition of electron, reduction of oxygen, and addition of a hydrogen is the reduction. It's very simple. If you remember these three over here, then it would be very easy to identify the chemical reaction. Adenosine triphosphate, actually first day when I talked about metabolism, I talked about it over here. So ATP or adenosine triphosphate, that's the energy currency of cell. Oh, I forgot to minimize it. Okay, so that's the energy currency of our cell. Means if you go to shop or store to buy something, you have to give money. Okay, so basically money is to exchange something, like instead you are paying money and the shop owner is going to give you something, your clothes or shoe or something. Okay, so over here inside cell, to buy something, cell needs to spend ATP. Okay, so it's like our money, our currency. So that's why it's called energy currency of cell. Structure, this is the structure of ATP, adenosine triphosphate. So tri means 3, okay, phosphate. So 3 phosphate group is there. So this is the 3 phosphate. Okay, adenosine, you know that AAT, adenosine, thymine, adenine, thymine, guanine, cytosine, you have already studied that. So that's the nitrogenous base. So that's the this is that nitrogen base. Okay, and that's the adenine structure. And this is actually 5 carbon sugar. So inside ATP, one ATP, there are 3 phosphate group, 1 5 carbon sugar, and 1 adenine nitrogenous base. That's the structure of ATP. Now, what happened when it is a phosphate? Phosphate is actually high energy molecule. Okay, over here inside ATP, 3 phosphate actually attached with a phosphate bond. These are the phosphate bond, and when we talk about that covalent bond, ionic bond, remember I told that bond means they store energy. Okay, so when in a chemical reaction, that elements or atoms, they make bond, means they actually store energy inside that bond. When they break down the bond, means they release energy. Okay, so that bond means that's the storage basically. So bond, you can say that's the storage of energy. Okay, so to make bond, we must have supply of energy, and when we break the bond, means we release energy. Okay, so if you remember these two, it will help you throughout the life. Very, very important. Making bond requires energy, breaking bond release energy. So over here in ATP, you can understand that 3 phosphate group, and inside there are 3 phosphate bond. Okay, so when they break this bond, they release huge amount of energy, especially when it is a phosphate bond between phosphate. That phosphate bond, they have capacity to store a huge amount of energy. So what happens that when ATP breaks down, let's say it can release 1 phosphate group, means when it has only 2 phosphates. So ATP release 1 electron. Okay, release 1 phosphate group means it actually releases a huge amount of energy, and then it converts into ADP, adenosine diphosphate. Then ADP can release another phosphate group and release huge amount of energy, and it can convert into adenosine monophosphate. Okay, so AMP cannot break anymore. But on the other hand, what can happen, that AMP can add 1 phosphate with the help of energy. It can convert into ADP. At the same time, ADP, if it can add up with 1 phosphate group, so it can convert into ATP. So actually utilize all these 3 chemical reaction whenever cell need it. Okay, based on the demand and supply. Okay, like in chemical reaction, it requires energy, so cell actually breaks down ATP to get energy. And when it produces energy, it has extra energy, then what it does, it actually converts all the ADP into ATP. Okay, for future use. That's how cell utilize ATP and ADP, and that's the reason ATP and ADP are very, very important for the for any cellular metabolic processes. Now, another thing I want to talk like phosphorylation. So over here, when ADP converts into ATP, what happened? ADP is going to add up with another phosphate, and then it produces ATP. Addition of phosphate group with any molecule is called phosphorylation. Okay, and when one compound, when they lose one phosphate group, that is called dephosphorylation. Okay, so these are the two terminology, very, very important: phosphorylation, addition of phosphate group, and dephosphorylation means reduction of phosphate group from in any element. Why I am talking about phosphate group? Because basically in the chemical reaction, when this exchange of addition and losing of phosphate occurs in the form of phosphate group PO3 minus. So that actually takes place in the chemical reaction. But this is not the chemistry, this is biochemistry. So we only consider the phosphate. Okay, so addition of phosphate with any element or atom, that's the phosphorylation, and losing of phosphate, that's the dephosphorylation. Enzyme, they are actually work as catalyst. They are the biological catalyst. So who are catalyst in chemical reaction? Catalyst are who actually speed up, it help chemical reaction it actually increase the speed of any chemical reaction. That's the function of catalyst. So catalyst, they speed up the chemical reaction. Okay, that's the one function of catalyst. And another characteristic feature of catalyst is they don't take part in the chemical reaction. Okay, they just help. So basically, when I'm telling that, okay, so they help to speed up the chemical reaction. Yes, they do. But at the end of the chemical reaction, they remain unchanged. So they actually do not take part in the chemical reaction. Enzyme is a biological catalyst. What they do, they actually speed up the chemical reaction, and at the end of the chemical reaction, they remain unchanged. Okay, and they don't take part in the chemical reaction, and that's why they are called biological catalyst. Now, every enzyme has two parts. Okay, let me. Okay, so enzyme has specialized area, and that area is called the active site. Okay, what happens? This active site. So this is, let's say, this is an enzyme. Basically, it has two specialized area, you can say. One is called the active site, and another specialized area, let's say over here, that's the regulatory side. Now, what happens, this active site and regulatory side. Active site for the substrate means reactants. I hope you remember the chemical reactions. So reactants, they together make the products, right? That's the chemical reaction. So like A plus B together, they make the product AB. So over here, A and B the reactants and AB is the products, okay? So that reactants has another name, and that is called substrate, okay? Now, this active site of enzyme is for these reactants or for the substrates, okay? In a chemical reaction, what enzyme does, it allow reactants to sit onto its body, okay? So that's the active side. And this regulatory side for other chemical molecules, elements, or compounds that actually control the activity of enzyme. Okay, so at this moment we will focus on the active side. So basically what it does, like how actually enzyme speed up the chemical reaction, how it works. So let's say this room is a pot or vessel where we are going to make water from hydrogen and oxygen. So just imagine it's a big vessel. Over here we have hydrogen. It's scattered. Okay, some hydrogen is here, other hydrogen is there, maybe at the down of the vessel, or a little bit of oxygen. Few oxygens are at the top of the vessel. So all the reactants like hydrogen and oxygen, they are scattered inside a big vessel. Okay, what enzyme does? Enzyme will collect all this hydrogen and oxygen on its active site. Okay, so to make water, you need 2 hydrogen and 1 oxygen. So 2 hydrogen, maybe from this corner, 1 hydrogen will come, another hydrogen will come from this corner, and from another corner, 1 oxygen will come, and then it can make water, right? And that takes time, okay? So it will take time for the reactants to come closer and to make products. But if you add enzyme, what it does, it actually, it has attraction, so it attract all the reactants into its active site. So, it saves time. So all the elements, if all the hydrogens are over here and all the oxygens are very close, so immediately when they are very close, they can start the chemical reaction immediately. And that's what happens when we add enzyme in a system or inside a chemical reaction. It immediately collects all the reactants on its active site and it saves the time. Okay, and this process is called, it actually lowered the activation energy. Means like when all the reactants are scattered, it requires energy to collect all those reactants together at one place. Okay, it requires energy, and that energy is called activation energy. Enzyme actually save that energy or reduce that energy by collecting all the reactants together. It saves time, it saves energy, and that's how enzyme work. Okay, and so this is the activation site. What happens when hydrogen, oxygen, or the reactants they sit? It helps those substrates to work together, and then it releases the product. At the end of the chemical reaction, the enzyme remain unchanged, okay? It does not take part in the chemical reaction. It just bring all the reactants closer. And that's how it reduces the needed energy or activation energy. Activation energy means to start the chemical reaction, how much energy we need. That's the activation energy. So basically, it reduces that energy, and that's how it actually saves the time also. It speed up the chemical reaction, but it does not take part in chemical reaction. So that's how enzyme work. So let me go back. So how do enzyme work? Enzyme actually lowering the energy of activation. It actually, to start the chemical reaction, how much energy it requires. If you add energy, you need less energy. And enzyme substrate interaction, so it brings all the substrate closure, and that's how it actually reduces the or speed up the enzyme. Induced fit mechanism. So as I told, that it has active site, okay, enzyme. So these two sites are the active sites, let's say for hydrogen and oxygen. These sites, they are very shape specific, okay. So what happens inside a cell? Lots of enzymes are there and lots of reactants, they move, okay. Only reactants like let's say hydrogen, it has a shape like this, okay. Oxygen, it has shape like this. Then only they will fit inside it. So that basically all these reactants, they are very shape specific, okay. So when this reactant shape actually perfectly match with the active site, so it's very easy going reaction. But sometimes what happens, that shape does not match. Like let's say now I have low sugar in my blood. Body needs to make sugar. So to do that chemical reaction, whatever the chemical reaction needs to start, for that, let's say what enzyme is there, that shape is not going or matching with the enzyme. In that case, what happens? But body needs that, right? So what body does, body actually send the that indication that we have to have this chemical reaction. So enzyme actually it moves a little bit, and it allow that substrates or that reactants to sit over here. So basically, based on, in emergency basis actually, based on that reactant shape, enzyme slightly change its shape to let that chemical reaction, which is very important at that moment inside the body, to happen, okay. So when this thing happens, that is called induced fit, okay. So basically enzyme actually adjust based on the emergency basis. And when just it goes regularly, like okay, so hydrogen oxygen together, at this moment they are going to make water, that's called lock and key mechanism. So it's like perfect match, so chemical reaction can happen. But over here it's not perfect match, but we must need to produce the products, okay. So in that case, enzyme actually adjust its shape, and that's called the induced fit mechanism. These are the two ways actually enzyme works. Now enzyme, I hope you remember that enzyme, that's a protein, okay. So when there is only enzyme, means when it is only a protein part, then that's called apoenzyme. But most of the time, only the protein part is actually inactive enzyme. They most of the time they cannot take part in chemical reaction. But if there is another element, means non-protein part, um, that is attached with this protein part, then enzyme become very active. Okay, this non-protein part and protein part together, basically when the enzyme is active, then that enzyme is called hollow enzyme. Okay, so only the protein part is called apoenzyme, but protein and non-protein part together make the hollow enzyme. Now I told that non-protein part again, it could be two different types. It could be inorganic or it could be organic. Okay, when that non-protein part is organic molecules, then they are the coenzyme. When that non-protein part is inorganic molecule, then that's the metallic cofactors. Okay, iron, copper, magnesium, all are the example of metallic cofactors or cofactors. Coenzymes means like vitamin B12. That's very important and very active non-protein part of enzyme. Okay, that vitamin B12 most of the time work with enzyme, and that's how we can absorb. Basically, it helps in assimilation of different nutrient elements inside the body. Enzyme inhibitors. So remember, I talked about two parts of enzyme. Okay, so one that's the active site. Okay, and another part over here or here, some other place. Okay, not the active site, where controlling molecules can sit on enzyme and they can control the enzyme activity, and that's the regulatory side. Okay, basically from where other elements can control the enzyme activity, that's the regulatory, or regulate the enzyme activity, that's the regulatory side. Now what happens when we do not need enzyme to be active? Like let's say hydrogen, oxygen together, they are going to make water. Okay, so but at this moment we have enough water inside our body. We really do not need to produce water. So what body actually does, body sends someone so that that enzyme can stop working, and it will stop producing or helping this chemical reaction, and that's how body actually control the amount of water inside the body. This is hydrogen, oxygen, water. I just gave you one example. For any chemical reaction, like when your sugar content in blood is too high, so body needs to stop producing more sugar, right? Or on the other hand, body needs to absorb the sugar from the blood. So that's the controlling mechanism of body. So in that case, what happens? That one of the regulatory molecule of enzyme, who are called inhibitors, who actually control the function of enzyme. They are the enzyme inhibitors. Okay. Now, two different types of enzyme inhibitors are there: competitive and non-competitive inhibitors. In case of competitive inhibitors, means they are the regulatory molecule. They are supposed to sit over here. Instead of sitting here, if they occupy that active site, means, so if they're sitting over here, they block the active side, and as a result, what happened? That hydrogen and oxygen, they cannot sit over here. So if they cannot sit, then they are not going to produce the water, right? So that's the competitive inhibitors. They sit they block the substrate molecules for the active site. They block the active site so that substrate molecule or the reactant molecules, they cannot sit on the active site, and that's how they actually control the chemical reactions. That's the competitive inhibitors. In case of allosteric or non-competitive inhibitors, they actually sit on the regulatory site over here. And once they sit over here, enzyme actually lose its shape. Okay, basically some chemical reaction occurs inside the enzyme's body, and it cannot allow the elements or the reactants to sit on the active sites. So that's the non-competitive or allosteric inhibition. So the difference between competitive and the allosteric inhibitors: competitive inhibitors, they sit on the active site. Non-competitive or allosteric inhibitors, they actually sit on the regulatory side, not on the active side. Feedback inhibition, like let's say, as I told, that you have too much sugar inside your blood. So inside our body, I think you had studied all these during AP2 and AP1. So body has its own mechanism. So when inside the blood, sugar level is too high, so brain, all the receptors particles, they are going to give the information to the brain, and from there brain actually send the information to a specific organs to secrete different types of hormone, and that's how then hormone will work on that organ or that system, and that's how body actually control or actually maintain the balance inside our body. So when it's too much sugar inside your blood, so brain will send information to the pancreas to secrete the hormone insulin. So insulin will take care of the excess hormone or low amount of hormone. So that's how actually that inhibition works. Okay. Now feedback inhibition is, that's the feedback, like body actually gives that information to the brain, and then brain actually take care of the situation. That's the feedback mechanism of body. Now inhibition means it actually, so one chemical reaction is going on, when it try to stop the chemical reaction. Okay, when body it form that, okay, so we have to stop the reaction, then that's the feedback inhibition. It is sending feedback to stop the chemical reaction. So that's the feedback inhibition. So over here I can so feedback inhibition, where the end product of the pathway serves as a non-competitive inhibitor or allosteric inhibitor to an enzyme early in the pathway. So what happens? So this is the chemical reaction. Okay, so basically this is the substrate, this is the active site, and this is the enzyme. So substrate will sit on the enzyme, and then it will produce one element, and then ultimately this is the end product. Okay, step by step reaction, and this will produce. So this is the starting point and this is the end point. But let's say this end product, this is the sugar, and at this moment let's say we have too much sugar, then body will send information to the brain. In that case, what will happen, that sugar molecule will sit on the regulatory site of the enzyme, and it will de-shape the active site, so that substrate will not fit into the active site, and it will control the production of that sugar. Okay, that's the feedback inhibition. So this is the end product. The end product will control or regulate the enzyme activity, and that's the feedback inhibition. So now the respiration, aerobic respiration. aerobic and anaerobic respiration. So when we inhale oxygen, okay, and exhale carbon dioxide, that's actually breathing, okay. That's breathing. So when you breathe means oxygen goes in and carbon dioxide goes out of the body. That's the breathing. Now, this oxygen, when enters inside the lungs, okay, from there, we have lots of blood arteries and blood vessels outside the lungs. What happened? They actually carry those oxygen to the different cell of your body. So all this oxygen, they entered inside a cell through the blood vessels, through the artery. Okay, and now this oxygen inside a cell, this oxygen, the presence of oxygen will help to break down the sugar into carbon dioxide. Okay, that's the respiration. So when breaking down of sugar occurs inside the cell, that's the respiration. And it is called cellular, and it happens inside the cell. So that's why it's called cellular respiration. Okay. Now, most of the animal, like for us, that we must have oxygen present inside the cell. So that's the aerobic respiration. There are few bacteria, instead of oxygen, they use nitrite, nitrate, all those. So in that means basically inorganic compounds. That's the anaerobic respiration. So when oxygen present during the cellular respiration, that's the aerobic respiration. When oxygen is absent, that's the anaerobic respiration. Fermentation, basically, it's the halfway of the aerobic respiration. It's a type of anaerobic respiration because the last part of the respiration, cellular respiration, does not need any oxygen. So that's why the fermentation is the part of anaerobic respiration. So there are these two types of respiration. Over here, this is the big picture of cellular respiration, okay? So what happened over here? This is the aerobic respiration in the presence of oxygen, anaerobic in the absence of oxygen, and what happened during fermentation. So aerobic respiration, it has 3 steps: glycolysis, Krebs cycle, and the electron transport chain. And in between, another step is there, it's called transitory phase. Okay, so this is one, glycolysis, first step. This is the second step. Krebs cycle, that's the third step, and electron transport chain, that's the fourth step. So there are 4 phases are there for the aerobic respiration. Now what happened? So first step is glycolysis. Okay, what happened? During glycolysis, that 6-carbon glucose, C6H12O6, actually breaks down into 2 molecules of pyruvate. Now that pyruvate or pyruvic acid, that's a 3-carbon compound. Okay, so from 1 molecule of glucose, we get 2 molecules of pyruvate. So the number of carbons, it's balanced now. Okay, so 6 equals 2 into 3 carbon. That gives us 6 carbon. So that's the thing happens over here. From 1 molecule of glucose, we get 2 molecules pyruvate. So, and during this time, a cell produces 2 molecules of ATP and 2 molecules of NADH. Okay, that's the net production. So that's the first step of glycolysis. So at the end of glycolysis, cell produces 2 molecules of pyruvate, 2 molecules of ATP, 2 molecules of NADH. Now this pyruvate converts into acetyl-CoA, coenzyme A, that is the transitory phase. Means in between the glycolysis and Krebs cycle, and that's why it's called transitory phase. And this time cell produces 2 molecules of carbon dioxide, okay? And this time, there is no production of ATP. Okay, so basically from one pyruvate, cell produces 1 acetyl-CoA, 1 carbon dioxide, but we have 2 pyruvate. So at the end of this transitory reaction, we have 2 carbon dioxide and 2 acetyl-CoA. Now this acetyl-CoA enters inside the Krebs cycle. It's a cyclic pathway, okay? Continuously happens, okay? So basically that end product of Krebs cycle actually works with the acetyl coenzyme A and ultimately produces carbon dioxide, ATP, NADH, and FADH. So 4 things produces carbon dioxide, ATP, NADH, and FADH. Okay, so over here 2 ATP is produced. Now what happened? So we already got ATP. So the purpose of the aerobic respiration is the production of ATP. So we produces 2 ATP over here, 2 ATP over here. So that is done now. But we need more energy. So what happens? All this FADH, NADH over here, they enter inside an electron transport chain. Why it's a chain? Because there are 7 large protein molecules. Okay, so these are the 7 large protein molecules. Okay, so that FADH and NADH, they start moving from one protein molecule to another protein molecule, and they actually jump from one protein molecule to another. And when they jump, they release electron over here. Okay, and that actually creates, it's called proton motive pores. Basically, the hydrogen ion concentration gradient actually creates over here. And as a result, there is another protein. It's called ATP synthase. It's a large enzyme. It's a motor-like structure. So it start rotating. And because of this movement of electron, and as a result, hydrogen ion concentration gradient actually creates. So this machine start working, and ultimately over here we have oxygen who accept all the electrons, and almost 32 ATP, 32 to 34 ATP is going to produce as a result of this electron transport system. Ultimately, at the end of the reaction, almost 36 to 38 ATP, carbon dioxide, and water is going to produce. So that's the aerobic respiration. So aerobic respiration means we have to have oxygen. So what's the function of oxygen? Oxygen, actually, they are the electron acceptors. So all these electrons, they are going to accept to neutralize the equation or the reaction. And that's how cell can make ATP from FADH and NADH. Okay, so that's the aerobic respiration. Now, anaerobic respiration up to this same. But during the electron transport system, oxygen is not there. Instead, we have nitrous, nitrate, carbonate, sulfate. These are all inorganic elements. They cannot absorb all these electrons. They cannot neutralize completely the chemical reaction. As a result, they cannot produce that much ATP, not 38 ATP. Sometimes they only can produce 2 ATP to 36, 30, at the max 34 ATP. But over here, no oxygen, so that's why the anaerobic. So steps are almost same. Only changes are here. Over here, electron acceptors are inorganic compound instead of oxygen. Okay, amount of ATP, aerobic respiration produces more ATP than anaerobic. Now fermentation, oh, my laptop it does not have charge. Just give me a second. So um, um, okay. Now fermentation, what happens? Only glycolysis occurs. Okay, so from here glucose breaks down into pyruvate, only that stage occurs. And we know from here to here, cell only can produce 2 molecules of ATP. So that's the production of ATP over here. Because after that, the pyruvate is not going to convert into acetyl-CoA and there is no Krebs cycle. So what happens, that pyruvate undergoes this fermentation process and it produces different type of acid or alcohol. Either it produces lactic acid or mixed acid. Basically, either it produces acid or the alcohol. Okay, so that's the fermentation. But during this time, cell cannot produce energy, ATP. Okay, so that's the highest production of ATP over here. Lots of bacteria, they do fermentation process. Okay, so that's the the differences among these three process. That's the big picture. So for glycolysis, every time glucose breaks down into 2 pyruvate, 2 molecule of ATP, NADH. During Krebs cycle, cells produces carbon dioxide, 2 molecule of ATP, NADH, and FADH. Fermentation, only glycolysis is there, and after that, pyruvate converts into either acid or alcohol. So if you understand that big picture, then it's pretty easy. So aerobic respiration, these are the 4 steps I already talked. So these are the steps. You really do not have to memorize all these, but just understand that over here, glucose, at this stage, glucose is converted into 2 molecules of glyceraldehyde 3-phosphate, okay? So basically, what happens from 1 molecule of glucose, we get 2 molecules of glyceraldehyde 3-phosphate here and here. Now, during the 3rd stage over here, we need to use ATP. Okay, and at the 1st stage also we are going to use ATP. So we use 1 ATP over here, and this is the 2nd ATP we use. So we use 2 molecules of ATP, like the ATP we had, we made it beforehand. So that stored ATP. Okay, so at the end, what happened? Glucose is completely breaks down into pyruvate. Okay, but at the 7th stage, ADP is converted into ATP, and at the 10th stage, ADP converted into ATP. So this is our first production of ATP. This is our 2nd production of ATP from 1 molecule of glyceraldehyde, but we have 2 molecules of glyceraldehyde, okay? So that we got from glucose, once glucose breaks down into 3 carbon compound. So basically 1, 2, 3, 4. 4 molecule, 4 ATP cell actually produced during the glycolysis, okay? So 4 ATP is produced, but we used 2 ATP. So net ATP production is 4 minus 2, basically 2. That's the net production of ATP during glycolysis. Okay, and we can see over here 1 NAD, NADH. So basically 1, 1. So 2 NADH. So what's the end product of glycolysis? 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate. That's the end product. And you have to memorize the end product. You really do not have to memorize the step, but you really need to understand that how the math, how ATP is utilized and how how it is going to produce, and how many ATP are going to produce, and what are the end products of the glycolysis. So that's the end product of glycolysis. 4 different types of process are there. This is the classic glycolysis we all do. Okay, so all the organisms, most of all the 99% of organisms follow this pathway. There are another one, hexose monophosphate. It's really old procedure, old organism. Now there are very few organisms who does that. This one, Ethnodeudrop pseudomonas, another one group of bacteria, like pseudomonas, who actually is responsible for your urine urine infection, and mostly ER infection. That's the pseudomonas bacteria. So they do this pathway. Phosphoketolase pathway, another group of bacteria, Bifidobacterium and Leuconostoc group of bacteria, they do this. But this is the most of the organisms who do this Enden-Meyer-Parnas pathway. Okay, so that's the transitional phase. What happened, that pyruvic acid is converts into acetyl CoA. Okay, so during this time, from one pyruvic acid we get 2 acetyl CoA, we get 2 carbon dioxide and 2 NADH2. That's the end product of transition reaction. Okay. So now we have carbon dioxide and NADH2. This acetyl coenzyme A or CoA, it will now enters inside the Krebs cycle. Okay. And what will happen? So the end product, it's a cyclic chemical reaction. Okay, lots of steps are there, but it continues in a cyclic manner. So the end product of Krebs cycle is oxaloacetate. That acetyl CoA that we got from a transitory reaction, that acetyl CoA reacts with oxaloacetate and it produces citrate or citric acid. So this is the first production during Krebs cycle, and that's the reason Krebs cycle is also called citric acid cycle. Okay, why citric acid cycle? Because this is the first production. Okay, citrate or citric acid. So what happens? So we get 1 NADH, 2 NADH, and so 3 NADH we get from 1 acetyl CoA, but we had 2 acetyl CoA. So ultimate production is 6 NADH from Krebs cycle. And remember, during ATP, during glycolysis, we had 2 NADH. So 2 NADH, now 6, 8, and transitory reaction, we had 2 NADH. So ultimate production, net production of NADH is 10 from all 3 steps. Okay. Now carbon dioxide, 1, 2. So 2 carbon dioxide means ultimately 4 carbon dioxide, because 2 carbon dioxide is going to produce from 1 acetyl CoA. Okay, so but we had 2 acetyl CoA, so ultimately 4 carbon dioxide. And remember, during transitory phase we had 2 carbon dioxide, so that's our net production. During cellular respiration, that's the total carbon dioxide production. Another thing happened, so ATP, only 1 from one acetyl-CoA means ultimately 2 ATP is going to produce. Okay. Another thing, new thing is going to produce over here, that's the FADH. So basically 2 FADH is going to produce from during the cellular respiration. So that's the net production of the Krebs cycle. So 2 ATP, 6 NADH, 2 FADH, and 4 carbon dioxide, that's the net production. So now if we calculate, remember, so from Krebs cycle, from one pyruvate we get 2 carbon dioxide, 3 NADH, 1 FADH, 1 ATP. So we have 2 pyruvate, right? So remember during glycolysis what happened? So glucose, 1 molecule of glucose actually breaks down into 2 molecules of pyruvate, right? So from one pyruvate during glycolysis, we Krebs cycle, we get this. So from 2, that's the production. So basically 4 carbon dioxide, 6 NADH, 2 FADH, and 2 ATP. Okay, and this process, like Krebs cycle and glycolysis, what happened over here? ADP is actually going to directly attach with the phosphate and it produces ATP. So addition of phosphate group, that is called phosphorylation, and it's the direct addition of phosphate with ADP. So that is called substrate level phosphorylation. So substrate level phosphorylation occurs in glycolysis and in Krebs cycle. Okay, and how many ATP we get from substrate level phosphorylation? 2 from the glycolysis and 2 from the Krebs cycle. So basically total substrate level production is 4 ATP. Electron transport, I will talk to that later. Okay, so I think we don't have time, right? Yeah. Okay, so let's keep up to this, okay? I will start from electron transport chain. I will do another video and I will post it tomorrow, so before your test, okay? So I will end this chapter and I will talk about chapter 9. So do you have any question for me?
Did you say you were going to have a video tomorrow, or was that Wednesday?
Tomorrow. I'm positively by tomorrow night I will post. So then you will have time to study on Wednesday.
Okay. So is it a Zoom meeting, or you'll just make it and then?
I will just go into Zoom and I will do it.
Okay. Is there a certain time or?
Hmm?
Is there a certain time?
No, I don't know what time I can do. I will do it by myself, okay? And I will post it. And just go over it. And if you have any question, just shoot me an email.
All righty. Thank you so much.
Okay.
Miss Nandy, do we have class on Wednesday over chapter 9?
I don't think so. If I post, the video of chapter 9, then we will not have a class. Then you can study by yourself. But
Okay.
in case I cannot do the video, then we will go over it on Wednesday.
Okay. And you're going to post it on D2L.
That makes sense. Like,
Yeah, that makes sense.
yeah. So because the test is going to open on Thursday, right? So I want to give you a little bit of time. I will try to post all the way up to chapter 9 by tomorrow.
Okay.
And then Wednesday you will be free to study.
All right. Sounds good. Thank you.
Okay. Good luck and study. And if you have any question, just shoot me email. Okay. Okay. Bye. Good night.
Bye.